Hello and welcome to the Select Science webinar titled Absolute Characterization of Glycoproteins and Their Interactions with Proteins and Antibodies by Light Scattering. My name is Anita Ramanathan and I'll be moderating today's presentation. I'm delighted to introduce our speaker, Dr. Daniel Sum, Principal Scientist at Wyatt Technology Corporation. In this webinar, you will learn how size-exclusion chromatography coupled with multi-angle light scattering helps characterize glycoproteins, aggregates, and complexes, and how composition gradient multi-angle light scattering can analyze self and heteroassociation in solution. Just before I hand over to Daniel, I want to mention a couple of things. We make this webinar as interactive as possible, so please feel free to ask any questions at any time by clicking on the Ask a Question button at the bottom of your screen. We'll have a question and answer session at the end of this webinar. Please be advised that along the presentation, a few poll questions will appear on your screen if you wish to answer them. You can also download documents in the Resources tab at the bottom of your screen. Now, without further ado, I'd like to invite Daniel and would like to thank him in advance for his presentation. Please join me in welcoming Daniel.
Thank you very much for that introduction. Great pleasure to be here today, and we'll be discussing the characterization of glycoproteins using light scattering, and also how light scattering can tell us about the interactions of glycoproteins with other proteins, whether they're target proteins for a biotherapeutic or if the glycoprotein itself is a target, and the interactions with neutralizing antibodies. The essential question we'll be discussing in today's webinar is how can I characterize this kind of a system where we have a glycoprotein, which has its essential protein molecular weight as well as degree of glycosylation, perhaps other self-association, interaction with the IgG with its own complications, and finally the formation of a complex between these two. What is its affinity? What is the stoichiometry? How many molecules of each protein come together to form the complex? So the way that we're going to address these questions is to use light scattering. Two techniques, which I'll discuss a little later on. They are called SEC mols and CG mols. We'll learn what those mean. And the question is, what can you learn about biomolecules with light scattering? So there are various light scattering tools that are utilized in biomolecular research. I won't be able to go into all of them today. I'll be focusing on the properties that are highlighted here in blue. So using light scattering, can we determine the structure and solution properties, in particular the molecular mass, the oligomeric state, and also the degree of conjugation or post-translational modification? And how does a protein function? How does it bind to another protein or to itself? Finally, light scattering tools are also used to determine if a protein that you have in hand is suitable for further research, and that's more of a quality control assessment to look at aggregation stability. And again, we won't be looking into that today, but if that does interest you, I would highly recommend going to the Wyatt website, wyatt.com/webinars, and we do have multiple webinars that address the uses of light scattering for some of these other applications. So what can light scattering measure? We're going to focus today on one particular type of light scattering, which is multi-angle light scattering. That's a form of static light scattering. And multi-angle light scattering essentially measures the ratio of the light which is scattered from a solution of molecules in solution to the incident light. And that will give us two quantities. The first is the molecular mass of the protein or of the molecule in solution. And second, its size in terms of root mean square radius or RG, also known as radius of gyration. And that happens through this first principles equation, which relates the amount of scattered light to the concentration of the analyte, a quantity known as the differential refractive index, the NDC, which essentially is the difference between the refractive index of the analyte and that of the solvent. And if we can measure the scattered intensity and measure the concentration and know or measure D and DC, then we can determine directly from first principles the molecular weight of the analyte in solution. You'll notice that there's one more term here, which is an angular term, p theta. And so we do measure multi-angle light scattering, which means the degree of scattered light as a function of angle. That will give us information about the size. But the range of sizes that we can measure with moles radii of about 10 nanometers and up are not applicable to many of the proteins you'll be working with, even to some of the standard complexes. On the other hand, if you're working on cage proteins or virus-like particles, those are actually large enough. They have RGs larger than 10 nanometers, and moles can give you their sizes, but that's not our focus today. There's another type of light scattering, which was well known in the protein world, called dynamic light scattering. And it also measures size. It doesn't measure molecular weight, but it does measure size through measurements of the diffusion coefficient, or the rate of diffusion under Brownian motion. And that can measure sizes which are relevant to most proteins, radii down to 0.2 nanometers. But again, we won't be discussing that today. Now that we know how to measure the molecular weight of a protein solution, we can also understand how it's possible to determine complete distributions of molecular weight of the monomers and aggregates and fragments and other things that may be in solution. Simply place the light scattering detector and concentration detector downstream of something that separates out the individual components, such as a size-exclusion chromatograph, and measure each eluting volume, and we'll get a complete distribution of molecular weight and size. Light scattering can also characterize conjugates that aren't really amenable to analysis by standard SEC or even by standard SEC moles. Conjugates such as glycoproteins, which combine protein components and a modifier such as glycans, don't have standard reference molecules and can't be analyzed by elution time. And they're also, for reasons we'll discuss later, difficult to characterize with a standard one concentration detector technique. And so it turns out that by combining 3 detectors, light scattering plus 2 concentration detectors, UV and refractive index, we can actually get the molecular weight of each component, the protein and the modifier, as well as the total molecular weight of the conjugate. We can measure conformation by comparing size with molecular weight. And we can also perform measurements of interactions using unfractionated solutions, which is known as batch measurements, by injecting into the detectors a series of concentrations compositions and determining how the molecular weight of the solution changes with these different compositions. So is it associating protein? Does the molecular weight of the solution increase with concentration, indicating formation of oligomers? Or if we mix together different ratios of protein A and protein B, do they form complexes, which will again impact the molecular weight? Light scattering can determine the binding affinity, Kd, as well as the types of complexes that are formed. So I'd like to begin with discussing SEC moles, or SEC moles, which couples multi-angle light scattering with standard size-exclusion chromatography, whether it's an HPLC system or an FPLC system. And the entire experimental apparatus utilizes an HPLC or FPLC with the UV detector from that system, and then adding in the moles detector, and also a refractive index detector, typically the white Optolab. So what we're going to do is we're going to use the HPLC to inject a sample, pump it through the column, separate out the species, and then those individual species will go into the detectors, the UV light scattering refractive index detectors. And then the software that analyzes the results is called Astra, and it also comes from White Technology. Inside the moles detector, what do we have? We have a laser beam, that illuminates a solution. And as the light impacts the molecules, it's scattered in all directions. We have a plurality of detectors that are placed at various angles theta relative to the direction of the laser beam. And by combining the information from all those detectors, we can determine the molecular weight and size. Okay. Why? So why do we need to go to all that trouble? We have a perfectly good HPLC or FPLC. Why do we need to add light scattering in order to analyze our proteins? Well, let's look at this case, which was published back in 1996 of an RNase. So when the RNase is in a buffer in which it's happy and it's globular and compact, it elutes relatively late, around 24 minutes here. But if you put it into a denaturing buffer, it elutes much earlier. So what does that tell us? Does it mean that the RNase is aggregated? Its molecular weight is much higher, and therefore it elutes much earlier? The answer is given by light scattering. So remember, the light scattering gives us molecular weight by looking at the ratio between light scattering intensity and refractive index and concentration. So that's basically the ratio between the height of this peak and this peak. And it's identical for the late eluting non-denatured RNAs and the early eluting reduced RNAs. They have the same molecular weight. So even though they eluted very different times, light scattering gives us the answer that they have the same molecular weight. It's been denatured. The reduced RNase has been denatured. It's unfolded, has much larger size, but the same molecular weight as the well-folded RNAs. In general, the reason that you need to add that traditional analytical size-exclusion chromatography and even native page can lead you astray is because they assume that the sample and references are very similar and ideal in the sense that they have the same conformation. So if your markers in page or if your reference standards in SEC are globular proteins, you're assuming that your sample is also a globular protein. It assumes that they have the same density. It assumes that they have the same interactions with the gel, and those interactions are perfectly steric. It ignores the option of having hydrophobic interactions or electrostatic interactions, which can change the elution time without changing the molecular weight. And they also assume reproducibility, that you're pumping through at the same rate and that your columns have not aged, et cetera, et cetera, et cetera. So all of these assumptions can be incorrect depending on the sample and depending on the chromatography system. On the other hand, when you add light scattering, it doesn't care about the elution time. As long as you can separate out the individual species and they come through the light scattering detector one by one, it will give you from first principles the molecular weight of each of those species, regardless of molecular conformation, how it sticks to the column, how the chromatography changes, independently of retention time. Here are two examples of protein analysis using secmols. And so the first protein, apelferritin, is a relatively compact protein that has a globular conformation. And we can see in red its chromatogram. So it has a monomer and a dimer and a trimer, and then some larger aggregates. The light scattering confirms for us the molecular weight of the monomer and dimer and so forth, showing that this is in fact the protein that we expect, but it also gives us some nice additional information, and that is that across the peaks of the monomer and the dimer, the molecular weight is quite uniform. So these are very homogeneous. There's no self-association going on. There are no additional impurities that are mixed in and perhaps eluting at different times. Quite uniform peaks. IgM is a much less compact protein, and therefore it has a larger volume relative to its molecular weight, and therefore its monomer and dimer do elute later on, earlier than for the apelferritin. Again, the light scattering gives us the molecular weight, confirms that this is in fact the monomer and that this is in fact the dimer, and also that the molecular weights are homogeneous across across these peaks. And the aggregates of both of these go all the way up above 10 million, so 10 and even 20 and 30 million Daltons in terms of their aggregates in this particular sample. What's interesting, though, is that the dimer of the apelferritin and the monomer of the IgM have approximately the same molecular weight, but they elute at very, very different times. And so if you're using standard column calibration, you would get very different results for the molecular weight. Mols is not fooled by the different conformations and does give us the correct molecular weights for both of these species. So that's how we analyze standard proteins. What happens with conjugated proteins, such as glycoproteins, lipoproteins, A membrane protein which is solubilized in detergent or lipids and actually might even be encased in a detergent micelle. So it's coupled to micelle of detergent. A nanodisk, which is a lipid bilayer stretched across a protein ring and might have a membrane protein implanted in it, and so consists of both protein and lipid bilayer. Other examples of conjugates are therapeutics, such as pegylated proteins. The PEG is used to increase the half-life of the protein in the body, or a polysaccharide protein conjugate vaccine. So these are all examples of proteins conjugated to something else. And how can we characterize them using light scattering? Well, we can't just measure their concentration using a single detector because they consist of two different species which have different UV extinction coefficients, different refractive index responses. So the answer is to use the triple detection system, which we show here below, consisting of UV detector, light scattering detector, refractive index detector. The UV detector will give us the concentration of the protein. In a typical example, such as a glycoprotein, where the glycans do not absorb UV. Refractive index, which is measured by the Optilab, is a universal concentration measurement, and so it will give us the concentration of the protein and of the glycans or the lipids that are bound to the protein. So between the UV and refractive index, we can figure out the concentration of protein and the concentration of the modifier. Combine that with the light scattering from the Mols detector, and then we will get the molecular weight of the protein, of the modifier, and of course then the total molecular weight, and that's shown here. So the conjugate analysis, which is performed in Astra, which is the software that runs these detectors, will give us results that look like this. We have a peak of a protein, and we can see that actually this peak is quite uniform. The modifier mass is uniform across the peak. The protein mass is uniform across the peak, and therefore the total mass is uniform across the peak, and we get all this information out of one measurement. Here's an example of using the triple detection system to analyze the glycosylation of a recombinant protein from two different host cells, mammalian and insect. And what we find is that the protein component in both cases is identical. It's the same protein, has the same molecular weight, is monomeric in both cases. On the other hand, the degree of glycosylation is different. The triple detection system shows us that there is more glycosylation in the mammalian cell and less in the insect cell, but it also shows us the heterogeneity of the glycosylation as we see the molecular weight of the glycans increase across both peaks in different ways. And so the triple detection system here gives us a look into both the protein content and the differences in glycosylation between these two different expression systems. SecMols gives us additional information about the behavior of proteins, and that is indication of self-association and the dynamic equilibrium of proteins that form dimers or higher oligomers in a concentration-dependent manner. What we see here is the injection of 3 different concentrations of protein. And if we look at the UV traces of the chromatograms, not only does the overall signal rise with concentration, but there's also a little bit of a shift. You can also see the broad shoulder on the right side of each chromatogram, which tells us that something is going on. It's not very clear. Light scattering, though, does give us the answer. It shows us that the molecular weight depends on the concentration along the peak, and it actually changes along the peak. It's not uniform, but it varies with concentration, both along a given peak and as a function of the overall concentration of the protein in that peak. And so here is a good indication of dynamic equilibrium. It's not highly quantitative. It's very difficult to determine from this type of analysis exactly which oligomers are forming, and it's difficult to extract KDs, but it is a great indication that we can then study further using CG-mols. So to summarize the applications of SecMols and also SEC with dynamic light scattering that I didn't talk about too much, they will determine the absolute molar mass and size of the solution components separating them out in SEC and measuring them with light scattering. They're essential for looking at non-ideal samples and non-ideal conditions where we might have column interactions, different conformations of the analyte relative to the reference molecules, and things happening in the chromatography, aging columns, et cetera, et cetera. It will determine the homogeneity or heterogeneity across a peak, which cannot be determined using standard column calibration. There's no way that you can know if a peak is homogeneous or not unless you use light scattering. And in particular, they'll help us to understand, in addition to the basic protein itself, how is it aggregated, how is it unfolding, what other impurities are present. For looking at conjugated molecules, it's essential to use light scattering combined with two more detectors, UV and refractive index, to look at glycoproteins, membrane proteins, and other conjugate molecules. Okay. So the second method that we're going to use using multi-angle light scattering is called CGMOL, stands for composition gradient light scattering, and it analyzes interactions. It uses the same detectors, the same multi-angle light scattering detector and UV detector that you use for SECMOLs, but instead of a chromatography system that's separating out the individual components, we're going to use the Calypso composition gradient system, which is a type of a stop flow system that combines up to 3 different solutions, injects them into the detectors, and stops the flow. You can wait for the sample to equilibrate, for complexes to form or dissociate, and then measure the weight average molecular weight of the solution and how it changes with concentration or composition. It uses different software, which is not Astra, it is the Calypso software. And this automated technique was pioneered by Atri and Minton back in 2005. It was my pleasure and honor to collaborate actually with Alan Minton on developing this system. So the world of composition gradient mols is very broad. It is a label-free and a mobilization-free technique for measuring protein-protein interactions. It will give you the binding affinity. It can also characterize non-specific interactions that don't have specific complex formation. It will give you, in addition to the molecular weight of the solution and the size of the proteins in it or the particles in it, the absolute stoichiometry. And I'll show you shortly what I mean by absolute stoichiometry. Essentially, if we have a self-association, is it forming a dimer? Is it forming a tetramer or something else? If we have a heteroassociation, is it 1-to-1, 2-to-1, or something more complex? CGMOLs can measure self-association, heteroassociation, simultaneous self and heteroassociation across a range, which does depend on the molecular weight of the proteins participating, but from below a nanomolar KD and up into the millimolars. And it works for all types of biomacromolecules, which scatter light, proteins, peptides, aptamers, whatever you like. I'm not going to go too deeply into the math. It's not really important for our discussion today. But what I would like to point out is that this is a first principles technique. It doesn't take into account the essential equations for mass action, mass balance, and the light scattering signal of a solution of proteins in dynamic equilibrium is composed of contributions that add up from the free monomers, which depend on the molecular weight and concentration of each of the free monomers, and the concentration and molecular weight of the complexes that form. So all of these equations scramble together or analyze simultaneously give us a highly constrained set of mathematical equations that will pin down the type of association that is occurring. So how do we measure self-association? Well, very simply using a concentration series. And so the Calypso system will mix up a series of dilutions. It'll start with a stock concentration at the maximum concentration and run a dilution series. Each one of these steps consists of a mixture which is injected into the detectors. The flow stops, as you can see in the step plateau here. The measurement is made. The molecular weight of the solution, the weight average molecular weight of the solution is measured, and the next injection comes in. So how can we analyze self-association in that manner? Well, if the protein stock solution is not self-associated, then its molecular weight will not change when it's diluted. And we'll see no change, if there's no interaction, in the molecular weight as a function of concentration. On the other hand, if there is formation of dimers or trimers or something else, we will see a change in molecular weight as concentration changes. At very low concentrations, we'll see a molecular weight that corresponds to that of the monomer. And as we go to higher and higher concentrations, we'll see an increase in molecular weight, which will, at high enough concentration, reach that of the dimer or the trimer or whatever else is going on. So the molecular weight will change with concentration, and also the form, the shape that this curve takes on of change in molecular weight with concentration will be indicative of the type of oligomer that forms. What about a heteroassociation? Well, normally when you think of a heteroassociation, you think of a titration where you have a fixed concentration of protein X and you titrate in protein Y and you get this sigmoidal curve, and the slope and the center will give you the information about the KD and the stoichiometry. We do it a little bit differently in CG models because each injection is a fresh preparation. We can actually address a larger range of stoichiometric ratios by doing what we call this crossover gradient, where we start with all of protein Y, measure its molar mass, then gradually reduce the amount of Y, increase the amount of X, measure a series of mixtures, and then finally measure just protein X by itself, measure its molecular weight. So if the proteins don't bind, the weight average molecular weight that we measure will just be this linear series from that of protein Y to that of protein X, which will look like this. But if a complex forms, the solution molecular weight will increase where we have the formation of complexes, and then we'll see this kind of shape. And the height of the peak will tell us about both the molecular weight of the complex that's forming and its KD. So the tighter the affinity, the lower the KD, the more complexes form, the higher the molecular weight of the solution for a given concentration. And so we can analyze these curves to determine KD. We can also analyze these curves to determine the stoichiometry or the absolute stoichiometry. So you'll notice that if we have a 1-to-1 or 2-to-1 or 3-to-1 complex forming, the peak of that complex formation will shift in accord with the optimal concentration that will give you, the optimal composition that will give you that complex. But you'll notice that the height of the peak also changes, and that's because we're measuring molecular weights. And so the molecular weight of 3-to-1 complex is higher than the molecular weight of a 2-to-1 complex, is higher than the molecular weight of 1-to-1 complex. If we have a 2-to-2 complex forming, so something composed of 2 X's bound to 2 Y's, it will peak at the same ratio as 1-to-1, but it will have twice the molecular weight as the 1-to-1 complex, and therefore twice the light scattering signal. And so again, by analyzing the shape and the height and the position of the molecular weight as a function of composition, these Calypso software allow you to determine the absolute stoichiometry of the complexes that are forming, and actually for systems that are much more complex than the ones we're looking at here. The software can analyze various types of interactions. So for self-association, you can analyze monomer-dimer interactions, or monomer-tetramer interactions, or even more complex interactions like monomer-dimer-hexamer. You can build fairly complex interaction models. Also isodesmic, which is a progressive self-association to form dimer, trimer, tetramer, pentamer, and so forth, assuming the same binding affinity for each additional monomer. There are heteroassociation models that can be used, so monovalent, 1-to-1. Multivalent, 1-to-n, with any n, assuming equivalent binding sites. But then there are also more advanced models. For instance, you can create models which are arbitrary combinations of self and hetero complexes. If you have protein A self-associating form dimers, and protein B self-associating form trimers, and A and B come together to form a couple of different complexes, all of this can be modeled up simultaneously. You can figure out which complexes are present in solution and which complexes that you might have actually added into your model are irrelevant and don't appear in the solution. You can analyze cooperativity, and you can also analyze, as we'll see later, some multivalent interactions where both proteins have more than one binding site for the other protein. Characterization of interactions by C.G. Mols gives you self-association of biomolecules, including the affinity and the oligomers that form. The solution-based analysis of heteroprotein-protein interactions, again giving you KD and the absolute molecular stoichiometry, not just how many moles of one bind with how many moles of the other, but actually how many molecules of each protein come together to form a complex. And because we're looking at molecular weights, and because we can analyze the entire gamut of associations that are occurring simultaneously, we get an intuitive and self-consistent interpretation of the complex interactions. So these are the basic tools, SCC mols, CG mols, and some other variants of light scattering that address the questions for biomolecular research that were brought up earlier on. So some basic characterizations first by mols. Here is the beginning of the analysis of a therapeutic glycoprotein. And what we see here are two peaks. So in the higher peak, we have the molecular weight of the protein, the molecular weight of the glycan, and we see that the ratio of molecular weights of glycan to protein is 0.13. And then this smaller peak has a ratio of 0.27. So it has about twice as many glycans per protein as the higher peak. Okay, that's interesting. Now what? Well, if we look at the larger species that are present in solution, we can see a few more peaks. We can see one which is the dimer of the high peak. Okay, so it's exactly twice the molecular weight, has the same ratio of glycan to protein, and we can find a trimer, which again has 3 times the molecular weight of the protein, and the same ratio of glycan to protein. Glycan to protein. So that tells us something interesting. We're getting aggregation of the less glycosylated protein, but we don't see any aggregation of the more glycosylated protein. And what that suggests is that the additional glycosylation somehow covers up perhaps some of the aggregation hotspots and is preventing the additional aggregation that the less glycosylated protein is subject to. Here's the analysis of a lactovirus glycoprotein with a neutralizing FAB. And this is work which was published not too long ago in Science by Catherine Hasty and her colleagues at Scripps Research Institute. And they were looking at the formation of complexes between the glycoprotein and the Fab using SEC mols. And we can see that the monomer itself in blue of the glycoprotein runs quite nicely and is quite uniform. But when you incubate the glycoprotein with the Fab, there is this formation of complexes, and the green trace shows the elution of that incubated solution. We have on the right side some unbound Fab here, because there was an excess of Fab, and so not all of it was able to bind to the glycoprotein. But the other two peaks do show complexes, and so the molecular weight of the middle peak, in fact, corresponds to the sum of the molecular weight of Fab and glycoprotein. In fact, these were analyzed using the complete triple detection system and the conjugate analysis, so we know that these are, in fact, properly glycosylated glycoproteins with the additional protein bound to them. And then the third peak on the left here actually corresponds to a trimer. So this incubation process brings about the formation of trimers of the glycoprotein Fab complex as well. And interestingly enough, these complexes are quite tightly bound, and even though they're run through the column and diluted and possibly even sheared, they do stick together. They do hang together as complete complexes and elute very nicely. There's a small degree of dissociation on the trailing edge of each one, but for the most part, they elute quite nicely as complete complexes. So now let's look at a case study, which is a viral glycoprotein, which exhibited both self-association and binding to a therapeutic IgG. And so this glycoprotein, because there were indications that it was self-associating, was injected with two different concentrations. The first concentration was a 100 microliter injection. It gave us a peak concentration of about 65 micrograms per milliliter. And at the peak, we measure 104 kilodaltons. And we also measure the degree of glycosylation and the molecular weight of the protein. And if you average them out, and the software gives this to you automatically, the overall DNDC, which is the contribution of both the glycan and the protein, is 0.177, which means that it's approximately 15 to 17% glycan. The second concentration was a larger injection. It gave a peak concentration of almost 200 micrograms per milliliter, so about 3 times higher, and it gave a higher molecular weight at the peak. Not only did it give it a higher molecular weight at the peak, which is indicative of self-association, you can also see a little bit of a dimer peak. So it looks like there is some dimer. It's not a very rapid dissociation. The dimer hangs together, at least some of it does hang together as it's eluting. Both of these have the same DNDC, so there's nothing going on in terms of different degrees of that glycosylation. It's the same glycoprotein, but slightly self-associating. Okay. Now we can analyze the self-association by CG-mols as well. And what we do is we use a method which includes both the concentration series, which is circled here in red, to look at self-interaction, and also the crossover series to look at the heteroassociation. So if we focus on the first part, which is the concentration series, we can see the molecular weights determined by CG-mols here in the blue diamonds, and the molecular weights determined by sec-mols here in the red circles. And we can see that there is a very distinct increase in molecular weight with concentration. It's not very large, which means that the KD is large compared to the concentrations that we're running here, but it is still there. So there is some formation of the tetramers. The essential molecular weight, as you extrapolate to 0, is in fact that of the dimer. So we do have a dimer with some weak self-association. Now we'll turn to the heteroassociation portion of the CG-mols measurement. And what we see is a series of injections, and for each injection, we see this drop in the light scattering signal, which corresponds to the early part of the injection where complexes haven't yet formed, so the molecular weight is low. And then as the flow stops, the reaction proceeds, the complexes form, the molecular weight increases, and the light scattering signal increases appropriately, and then finally reaches equilibrium. And we'll be taking data from the equilibrium region of each of these plateaus. And so here are the results of this analysis. The blue circles here correspond to the equilibrium data taken from each of the compositions shown here. Now, if there were no interaction between the molecules, then we would expect to see a series of points which run along this dotted line, and that's the no interaction line. Obviously, that's not the case. We can also try to fit the data to a model that consists of a 1 to 2 interaction only. That's the dashed green lines, and that's a model that assumes that the IgG can bind up to 2 of the VGP dimers. And you can see that it approximately fits the data, but it's not a very good fit. There's something missing. In fact, in order to get the great fit that we see here in red, we need a model which includes 3 species. The 1-to-1, 1 IgG to 1 VGP dimer, and 1-to-2, so 1 IgG to 2 VGP dimers with equivalent binding sites. So the same association constant for each of the VGP dimers to the IgG Fab arm, and also a term which includes 2 IgGs binding to a VGP tetramer. So remember we saw both in CG-mols and in sec-mols that there's a formation of a little bit of VGP tetramer. That tetramer is binding to IgGs, okay? And so what tells us, so overall, if we look at what we've learned from VGP and its interaction with the map from CG-mols, is that it behaves as a native dimer with a weak self-association. The VGP dimer has only 1 binding epitope for IgG. IgG can bind 1 or 2 of the VGP dimers, but one VGP dimer cannot bind two IgGs, and the KD that we've determined from the analysis is 58 nanomolars for VGP dimer to IgG. Finally, we do need, in order to get a good fit, an additional term, which is a higher order complex consisting of a VGP tetramer, which has two epitopes now for IgG, and it's binding two IgGs. And that's that extra blue weight, which is given over here. So this is where we have the self-consistency. We found that there's an IgG tetramer. We expect it to be interacting. We find that it interacts, that it binds two IgGs as we would expect for when the dimer has a single epitope for IgG. The final case study that we'll be looking at today is the analysis of an interaction of an Ebola glycoprotein with a therapeutic IgG. And this material was taken from a webinar presented earlier this year. If this is a topic of interest to you, I definitely recommend viewing it on our website. It was presented by myself together with Catherine Hasty of the Ullman Sapphire Research Group at Scripps Research Institute, where this is a primary topic of research for them. And SECMols and CGMols figure heavily in their research. The Ebola surface glycoprotein is trimeric, and it was tested against a variety of therapeutic IgGs. The one we'll be looking at specifically is IgG 13Z6, which binds to the glycan cap of this protein. In the interest of time, I'm going to cut straight to the final analysis of the CGMols data for this interaction. And here's what we find. We find a very high abundance of the one-to-one complex between the Ebola glycoprotein and the 13C6 IgG. So we do have a one-to-one interaction. Oddly enough, as we can see in the lower left-hand corner, there's no evidence at all for that same IgG being able to bind two of the glycoproteins, one on each of the Fab arms. We do see evidence for a smaller abundance of two IgGs binding to the trimeric glycoprotein, so not surprising that it can bind, that the trimer can bind two of these IgGs. And then also a small abundance, which in terms of mole fractions it's small, but in terms of light scattering it's fairly significant, is the 3-to-2 complex, which combines two of the trimeric glycoproteins with three of the IgGs, and this is one of the fun things that happen when you have multivalent interactions. Even stranger, we find an unusual distribution of KDs. So the one-to-one interaction is very tight. It has a KD of 750 picomolars, less than 1 nanomolar. On the other hand, if we add another IgG to that complex, that one-to-one complex, the additional IgG binds weakly with just 300 nanomolar KD. And the 3-to-2 complex, if we average out the KD per binding site, it's somewhere in between. It's about 14 nanomolars. So there's a large difference in affinity between different complexes. Now, there are a couple ways that can happen. There can be cooperativity. In this instance, it looks like negative cooperativity. There can be other effects that happen. There's this absence of one IgG binding two glycoproteins. So all this tells us that something about our hypothesis may be incorrect. And perhaps the geometry is incorrect. If we want to understand the geometry, go to cryo-EM. And so this cryo-EM image also came out of the Ullman Sapphire lab. Was published in Nature Microbiology together with some of the CGMols results. And what it tells us is that this antibody actually binds with both Fab arms on the same glycoprotein. Both of the arms of the antibody, both of the Fabs, will bind to the two epitopes on the glycoprotein. Well, now things are a bit clearer because it tells us that the revised geometry for the one-to-one is not a single Fab arm bound to a single glycoprotein in the trimer, but it's actually two. There's an avidity effect where even though the interaction of one arm with one glycoprotein may be weak, Once we have 2 bound, it becomes quite strong. And that explains why the KD is so strong for the one-to-one interaction. Now, when we get down to the 2 IgGs bound to the 1 glycoprotein, well, if two of the glycoprotein epitopes are bound up with an IgG, that leaves just 1 free. So if we have an excess of IgG, it can come along, but it can only bind the 1 remaining glycoprotein epitope. And so that's going to be a weak interaction, just 300 nanomolars. Then putting this all together, it tells us, well, the third species that we saw was actually 2 of the one-to-one complexes, which are tightly bound because both Fab arms are bound to the glycoprotein. And some additional IgG came along and was able to bind weakly the remaining epitope from each one of those, and that creates the 3-to-2 interaction. And so the average interaction here is somewhere in the middle. It's about 14 nanomolars. The beauty of CG-mols is it gives you all this information. We see here some ITC data on the same system, and ITC unfortunately doesn't give you all that information. It will give you an average KD, which came out to about 80 nanomolars here. It'll give you an average ratio of IgG to glycoprotein, which is about 1.3, so somewhere between 1.1 and 2 to 1 and 3 to 1, 3 to 2, somewhere in between. But there's no way that it can distinguish all the different complexes that form, and so it just gives you just an overall average. CG-mols really gives you the complete. So why use light scattering to characterize glycoproteins? Well, sec-mols will give you the determination of homogeneity, the oligomeric state, and the glycan content. CG-mols will characterize the interaction of the glycoprotein with its target, or how a neutralizing antibody interacts with the viral glycoprotein, so that we can understand the absolute stoichiometric models that form the actual complex that form in solution, and more fully characterize the therapeutic candidates. If we add in some additional techniques, and in particular cryo-EM here was very useful, it helps to flush out the picture that CG-mols gives in combination. They synergize and provide a complete picture of the complex interactions. So with that, I thank you for your attention. If you're interested in more information, here's some links to learn about sec-mols, CG-mols, some additional webinars on these topics. And of course, you're free to contact me or info@wyatt.com for additional questions. And again, I'd like to thank Dr. Hasty for sharing some slides. And with that, I'll return it to our host, and I'm open for any questions.
Thank you, Daniel, for your informative presentation. Now let's move on to the final part of today's webinar, the question and answer session. We have lots of great questions, so let's begin. The first question is, which type of pump is used for the CG-mols? Daniel?
Yeah, so the Calypso composition gradient system actually uses 3 pumps. They're computer-controlled syringe pumps, and by operating them in such a manner that they produce different ratios of speed, they will combine different compositions of the 3 solutions.
All right. Thank you. The next question is, what additional information can you gain for the concentration-dependent SEC mols when you add in the RH and retention time data? Is the data simply confirmatory of a complex dynamic, or is there further granularity that can be gleaned?
So both RH, the hydrodynamic radius, and the retention time, assuming purely steric interactions, is also indicative of hydrodynamic radius, can be combined with the information of molecular weight to get a feel for conformation. If the protein is purely globular, then there's a known relationship between hydrodynamic radius and molecular weight. But if the complex it forms comes together in ways that produce a more extended configuration, that will show up in the hydrodynamic radius information as well.
All right, thank you. The next question is, can SEC mols with UV and RR detectors analyze small glycoproteins? The protein sequence weight of my samples is about 10 kilodaltons, and I expect that the total glycan molecular mass is about 1,000.
Yes, no problem at all. secmols in general can measure down to even 1,000 daltons. In terms of measuring the degree of glycosylation, what we're looking for is at least 3% in mass of the glycan relative to the protein. So in your case, where the glycan is about 10% of the protein molecular weight, that should be straightforward to measure using SEC mols with UV and RI.
All right. Next question is, what is the minimum degree of glycosylation that SEC mols can quantify?
Right, so this ties into the previous question, basically 3%. So mols in general has a degree of repeatability and precision on the order of 1%. So we're looking for about 3 to 5% in order to get a good measurement of the degree of glycosylation.
All right, the next one is, I only have 10 micrograms of protein for analysis. Is SEC mol suitable for such small quantities?
It is in general. So even if you're using standard SEC with typical GPC columns, you can measure that quantity of sample. In fact, we can measure down to less than a microgram injection. But really, the better way to go is to use SEC with mols with UHPLC. So we do have a UHPLC compatible mols detector called the microdom, and it will combine with UPLC and readily measure that much material and even much less. Actually, you can go down to 100 nanograms and below.
Thank you. The next question is, how do CG mols results for KD compare to other techniques such as SPR or analytical centrifugation?
So these have been tested in a variety of situations, and in general, what we find is that whenever you're looking at KD measurement in solution, the CG mols results are always comparable and very similar. In fact, the technique itself was validated against analytical ultracentrifugation, in particular sedimentation equilibrium, which really is the gold standard for this type of measurement. When you're comparing to SPR, you have to be careful because now that the proteins are no longer in solution and one of them is mobilized, that can impact the interaction in general. So often we see that where the measurement in SPR is good and is comparable to other solution-based measurements, the CG mols measurements are very similar, except that CG mols actually also gives you, of course, the stoichiometry in solution as well. But sometimes immobilizing protein on the SPR chip will cause the interactions to change, and that will be reflected in a difference between the two measurements.
That's great. Thank you. The next question is, how much protein does CG mols require?
It depends to some degree on KD. Of course, the higher the KD, the more protein you need. But for typical measurements with KDs on the order of 100 nanomolars and below, it takes about 100 micrograms of each protein to make the measurement. If the interactions are weaker than 100 nanomolars, then it will require more protein.
Okay. The next question is, can protein used in SEC mols or CG mols analysis be recovered?
Absolutely. These are non-destructive techniques, and especially if we're only looking at a single species and, say, measuring self-association and dilution, we can recover that protein, reconcentrate if necessary, or use as is. Nothing's happened to it. And if it's come through the SEC mols column and it's not impacted by the column itself, the detectors downstream don't impact it very much. Fractions can be collected, or pools can be combined, and so you can reuse the protein. It's perfectly viable.
That's great. That's all that we have time for today. Thanks, Daniel, for your discussion and the presentation, and thank you, our attendees, for joining us online and for your interesting questions. I hope that you found this session valuable. If I didn't answer your question today, please feel free to email me at anita.rama@selectscience.net, and I'll personally follow up with Daniel. We'll soon have an on-demand link for this webinar in a few days, so you can listen to it again or share it with a friend. Once again, thank you for joining us, and I'll see you next time. Thank you.